Hi guys, Dr. B here. So this is the rest of chapter 6. It's about 9 to 10 slides. We'll see if we can get this within the 10-minute mark. So we're going to start this last section with the 5 phases of the cell cycle. They are the G1 phase, S, G2, mitosis, and something called cytokinesis. So G1 phase refers to rapid cell growth. The S phase is when DNA is copied or synthesis. G2, microtubules are rearranged to prepare for mitosis, which we'll get into the specific steps for this as well. Mitosis is basically the nucleus of a cell is divided into 2 nuclei, and cytokinesis is cytoplasm division. Some cells actually remain in G1. They just grow and grow, cell growth. They don't divide. So that is the muscle cells and your nerve cells. They do not really create, they do not divide. They actually stay and they just keep on growing. Mitosis and cytokinesis allow organisms to grow and replace damage because they're replicating new cells. So looks a little confusing. So this is the cell cycle. Let's focus on G1. So that's growth and normal metabolic roles. And then you have DNA replication in the S phase or synthesis phase. And G2, which is growth and preparation for mitosis. Excuse me. And you know that, well, you'll know in mitosis, you'll have the 4 steps, which are going to get into more detail in the next couple of slides. So there are certain checkpoints within the cell cycle that allows for, how should I put this? Basically, it's making sure everything is okay, and it's catching mistakes as the cells are replicating. So the first one is the mitosis checkpoint, which signals the beginning of G1. In the G1 checkpoint, there's something called a restriction point, and at this point, the cell becomes committed to entering the cell cycle. The G2 checkpoint involves DNA repair enzymes, rapid growth, and protein synthesis in preparation for mitosis. This term right here, interphase, anytime the cell is not dividing. So if you can see in the actual diagram, it shows that G1, S, and G2 are all part of interphase when the cell is not dividing. Hint, hint. So when control is lost, this is when you can end up with cancer. Well, there are different types. So if I just say cancer, it's a scary word, but there are different types that you have to kind of keep in mind, which we'll get into as the school year continues. So what do you call it when your sister steps on your toe? What do you say? You say, ouch, my toe, sis. Get it? Yeah, I know. Nobody's laughing. It's fine. No big deal. Alrighty, so chromatid separation. So chromatids on each of the chromosomes are physically moved to opposite sides of the dividing cell with the help of a spindle. A spindle is a cell structure made up of centrioles and individual microtubules that help move chromosomes apart during mitosis. So, which organelle is involved in cell division? One of y'all described this perfectly when we had our our jigsaw activity with the parts of a cell. So cell division is centrioles. That is the organelle that's involved with that. So mitosis under a microscope. So this is what it looks like. So prophase, metaphase, anaphase, and telophase. These are all the 4 steps of mitosis. So in prophase, chromosomes become visible, and the nuclear envelope that houses, within the nucleus dissolves. The spindle forms. Metaphase, chromosomes line up along the equator. They line up along the middle. Anaphase centromeres divide and the chromatomes move apart to opposite poles. This is where the centrioles help. So anaphase kind of looks like A's going that way, if you will. And we want to erase that. Make it a different color. Let's go yellow. Anaphase, they're making sideways A's going opposite directions. Then telophase, the chromosomes uncoil, and you're going to end up with 2 nuclei. Notice how this is more in depth than, say, binary fission. Just kind of keep that in mind. When telophase happens, or when it occurs, when it's finished, the spindle dissolves and you end up with cytokinesis, and that is division of the cytoplasm. The result you get from cytokinesis is two genetically identical cells from only one cell. So in terms of what happens in cytokinesis, you have cells without a cell wall and cells with a cell wall. So this one is, to simplify it, we're going to say this is animal cells, and these are plant cells. So we're going to describe them, and you're going to see a microscopic image of what it looks like. So without a cell wall, cells are pinched in half by a belt of protein threads. You can see it kind of does like whoop. With a cell wall, however, vesicles from the Golgi apparatus fuse at the midline and form a cell plate. A new cell wall forms on both sides, and then the cells separate. See how it's a little bit different? You can see the cell membrane here. However, here you can actually see an actual cell wall, and boom. So that is it for chapter 6. We will continue with chapter 7. If you have any questions, let me know. Hit the like and subscribe, and I'll see you in class.